Now you can all see that? Great. Yeah, so my name is Joseph. I was a PhD student in Zoe Cortzi's lab in the Department of Psychology. I actually, when I give this talk to data scientists, I tend to talk about neuroimaging. And when I give it to neuroimagers, I talk about kind of the methods. And now this is kind of the perfect group, but I'm in a MRI session talking about PET. So I think that maybe it's me that's broken here. But this is going to be kind of a case study in this method and how we've applied it. So I'm going to keep things quite general. So if anything kind of sings out to a similar problem you have, then you can use any type of data you like to kind of make these predictions. So the Alzheimer's disease cascade, for people who aren't familiar, is kind of this transitional cascade of different pathologies becoming more predictive of where you are in terms of disease stage. So in this curve, we see the kind of clinical diagnoses, and it changes from cognitively normal to mild cognitive impairment to dementia. And throughout these changes in clinical diagnoses, you see a change in biomarkers. So for instance, when people are cognitively normal, they're starting to accumulate amyloid, which is a key protein in the Alzheimer's cascade. And so you see that while there might be these broad diagnostic categories, there really is a lot of continuous information in biomarkers that's happening kind of under the hood of the patient, so to speak. And really this approach goes at how we can kind of pull that information out when we're just trying to train a model using these diagnostic categories. So the machine learning approach that I use is this GMLVQ. And so this is kind of it's a geometric way of performing a binary classification. And I'll just go through how this works kind of graphically and also in terms of a higher order example. So let's just first define our classes. So if we're going to train a classifier using baseline data, we want to try and say who is going to progress to dementia due to Alzheimer's disease from this kind of interstitial stage of mild cognitive impairment. So the problem becomes like this. Of our 4 training samples, 2 will retain a stable diagnosis of MCI, and then 2 will progress to dementia. So now what we've got, if we've got our classes assigned this way, we've got 2 features. It can be whatever you like. And this is what these data points look like in this hyperdimensional space, in this theoretical or example given here. And so what we're trying to do is use baseline predictors to separate these people based on their future diagnosis. So the first step is we're going to try and scale our data so that points of the same class are closer to each other. So we want the red dots to be adjacent, the blue dots to be adjacent, and the red and blue to be far away. And the first stage that happens is you learn this metric tensor. And that's just going to scale your feature space to try and force these data points together. So all we have to do to now create this transform space is just times any new data point by this metric tensor. Now, when we're in our learnt space, we want to say how close are we to each other's points? And we want to find kind of the median of this distribution, or the prototypical location for someone who's of class, unstable diagnosis, so they're going to convert to dementia, or someone who has a stable diagnosis. And so we can just define that point in our learnt hyperdimensional space. Now, if we have our test point, again, this is all baseline data, we just scale the data to start with, and then we just see where they fall in reference to these prototypical locations. And so in the binary sense, we would just say that this person has an unstable diagnosis based on their baseline data. So this is the binary classification problem. Except what we wanted to do was try and pull some more information from this trained classifier and make some continuous predictions. And so let's go back to before we classify our individual. We've got our test point in this learnt space, and we've got some locations that kind of define our training sample. Now what we're going to do is we're going to define in our learnt space this vector, or this prognostic axis. So what it's saying is along this dimension, this hyperdimensional space, this is what separates people who are stable and people who are unstable in terms of their diagnosis. Now what we do is we just project up our sample point along this vector. And what we get is a scalar number. So this blue line, or the length of this blue line, is effectively how far away our test point is from the healthy individual along the axis that is predictive of change of diagnosis. So now what we've got is, because we're using baseline multimodal data, we've got this projection or a continuous multivariate index, which is going to say how far away someone is from a prototypical location. So back to kind of the problem with Alzheimer's disease. As you see this transition from mild cognitive impairment to dementia, there's biomarkers that are changing kind of underneath the hood. So you see that memory is changing and also atrophy is changing during the transition between these two diagnostic categories. So if we look at the purple line in these curves, we see that, yeah, memory is changing. And so what we're going to do is use our scalar projection to try and predict that continuous change in memory. So using baseline data, we trained our classifier with structural MR, so a metric of medial temporal atrophy, total amyloid burden across the brain measured with PET, and APOE4 genotype. And our continuous metric we want to predict is the future rate of memory change. So that's from their baseline data that's used to train the model. How are they going to change their memory going into the future? And what we do is we take this scalar projection, we show it on the x-axis here, and then we just relate it in a regression with the future rate of memory change. And so we saw in this sample that we could predict close to 50% of the variance, and this is actually in a holdout data set. So these individuals weren't used to train the initial model. So that was kind of the first project we did to try and predict these continuous trajectories. Now we wanted to go to a newer project where we're looking at earlier changes. So again, we can define our groups this way of people who are early Alzheimer's disease, and they are the cognitively normal MCI, and they will progress to dementia, or people who are stable cognition. They'll just stay cognitively normal into the future. And again, what we're going to do is we're going to use the same three baseline multimodal predictors. So structural MR, this medial temporal atrophy score, total SUVAR, and APOE4 genotype. So to start with, we wanted to see how well we could classify these groups just using a binary classifier. And to do that, we had to decide where a decision boundary would lie using this scalar projection. So we just fit a logistic regression to the scalar projection, which is the x-axis here. And we could find that there's a boundary that exists that can separate the two groups, the stable cognition and the early Alzheimer's disease, with pretty good accuracy. Right, and so what's the continuous metric we wanted to do from our trained classifier? And so you see in this transition from cognitively normal to MCI to dementia, that you're seeing the accumulation of tau across the brain, which is another characteristic protein in Alzheimer's disease. So the reason why this approach is actually quite good is because our initial training sample, they're from a database called ADNI, but they were kind of collected in 2010 to 2013, these first training samples. And this actually predates the widespread uptake of the tau tracer that's used now in PET. So what we did is we trained our initial model using these subjects. And then now for two different samples that have longitudinal tau, we calculated this scalar projection and tried to see if we could figure out how this related to future tau for these different individuals. So what we did, again, out of sample, is we calculated this future change in tau accumulation across the cortex, and we just contrasted these two groups. So the people classified as stable cognition, who are on the left, and they actually have barely any tau accumulation across the cortex, with some in the medial temporal lobe. And then with the people who are classified as early Alzheimer's disease, you see that they do have this kind of Alzheimer's topography of tau accumulation across the cortex. And looking on average, these people were accumulating tau 2.8 times faster. So the next thing we could do is we could look within these ROIs and say, are they significantly accumulating tau? And so we see within the temporal and parietal regions that for the early Alzheimer's disease group, that they are accumulating tau. So we wanted to then test, can we do a continuous prediction within these regions of interest? So if we average the future tau accumulation across parietal regions, as well as within the temporal regions. And so showing here, we then fit this scalar projection on the x-axis and the future rate of tau accumulation in these temporal and parietal aggregates. And so we see we can fit this continuous rate of change in tau accumulation in a holdout sample effectively, and that it does kind of predict across both the temporal and parietal region. But what we can also do is we can effectively regress this metric across the whole cortex, and we can get a series of regression equations for each of the ROIs. And we see again that we can predict regionally quite specifically. So you see different distributions within the temporal cortex and within the parietal cortex, where you can use this metric to predict future tau accumulation. And now we've got a second holdout sample. We wanted to see of this cognitively normal community dwelling population, which is this Berkeley aging cohort, can we fit them into our regression equations from ADNI3 and try and predict completely out of sample? And so this is within these aggregate regions where we fit the Berkeley aging cohort into the ADNI3 regression equations. And we see that we can fit a reasonable model and we can predict a reasonable amount of variance within these two areas. And then, of course, we can plug these data into the whole brain's regression equations. And we see again across the cortex, we can explain a fair chunk of continuous variation, up to about 40% in some temporal regions. So what's the benefits of this type of approach? This classifier is actually well suited to handle correlated predictors. So if you look at this hypothetical scenario in the plot here, the things that separate blue and red are correlated. So an increase in Y and an increase in X will separate the groups. And this correlation gets modeled by the off-diagonal terms in this metric tensor. So this term will collapse this angle and improve the classification. The second and arguably the most important thing of this approach is that we can extract continuous information from discrete classifiers. So why is this important? So from the idea of transfer learning, these relationships that they're uncovering in change and pathology is implicitly learned. And so if these relationships are implicitly learned, we can train the sample with missing data and make predictions in data that's collected in a different sample. The third thing is that we can train a model with high target uncertainty. So it's a bit of jargon, but what it's saying is when I build my binary classifier, my labels that I'm giving it to learn, they might have a bit of error in them. And this is important in this type of a model and this type of a problem where you're trying to classify changes in diagnoses. Because the reason why someone might change has to do with a lot of different biases in the actual sample itself. So for instance, if I've got a study that varies its follow-up, so my future could be 3 years, but I've got another study that has a follow-up of 3.5 years, then you'll see that my problem will change quite a lot. And this point here might actually transition into a different group. The second thing is that clinical decisions are generally quite hard in this problem because they rely on quite a lot of information that you don't get to see. So for the clinician that decided here 1 day earlier, why has this happened? That's a tough thing for the model to try and learn when you give it these types of input variables. So by not forcing it to try and replicate a clinician, you kind of leverage its ability to extract better information and more predictive information. I'd like to acknowledge all the people that I worked with here in Cambridge and at UC Berkeley, and then the computer science team at University of Birmingham as well. There's 2 publications. One's in revision and should hopefully get through soon. And yeah, please email me if you have any questions or ask some questions now.